Welcome back to the course on fluorescence spectroscopy. We're going to begin our discussion of fluorescence lifetime imaging microscopy. This was sort of a miracle when it first came together because it seemed like it'd be something almost impossible to do. But now it's present on many microscopes. Of course, it can be done both in the time domain and the frequency domain. We'll start by discussing the time domain measurements because they're easier to understand. So what's the objective of FLIM? We want to get an image of the sample that is based on the chemical composition and not on the amount of probe that's in any one place. And what I mean by that is you will see in the literature many beautiful pictures of cells. They're stained and usually they're fixed as well. And these are cells that have been stained with different small fluorophores, antibodies labeled with different fluorophores, almost for the purpose of getting a nice picture on the front of a magazine. This one you can tell nuclei are stained with DAPI. Some biomolecule here is stained with fluorescein, another one is stained with rhodamine. They're beautiful images, but they're not always the information that the biochemist would like to have. So what's our expectation from the FLIM measurements? So let's consider a cell that has two different regions in it. And this picture has calcium on it because when we discovered FLIM, calcium was one of the things that everyone was trying to measure inside cells. And if you label a cell with a calcium-sensitive fluorophore, what you're likely to do is just see an intensity that is proportional to the amount of probe in that particular location of the cell. And that's not what you want. What you wanted to see is how much calcium is in each location. So that's what we call FLIM. We create contrast, which is based on the amount of the analyte that's present. So it's an artificial contrast. It's a false color image, but the images tell you, there's so much of this species here, or there's so much of this protein that is undergoing fluorescence resonance energy transfer. To the best of my knowledge, we were the first group to understand that FLIM was possible. When this idea first came out in the 1990s, it was really considered almost impossible. In fact, I remember we were waiting for a site visit from the NIH, and we had proposed FLIM, and we hadn't done it yet, and we knew if we didn't get images by site visit time, we would be dead in the water. But lo and behold, we got the images, and that helped us get the award to further develop FLIM and other things. But prior to 1990, this was really considered almost impossible. Remember when we spoke about the flash lamps were unstable, the repetition rates were in the kilohertz range, and remember you can only take 1 photon per 100 light pulses to avoid pulse pileup. So even doing a low resolution image, 256 by 256 pixels, it was really an impossible task. So you're never going to do that. But we thought from some of our previous measurements that it would be possible with the frequency domain or phase modulation measurements, which we will discuss in the following lecture. So our motivations were to reveal local concentrations, the state of the probe. The there's always photo-bleaching and phototoxicity. We had just undergone a study of looking at a number of ion-sensitive fluorophores. So this was a motivation for doing point-of-care testing. So we knew that many of the ion-sensitive fluorophores had lifetime changes, and they could be used for FLIM. So this sort of we had some recent observations that suggested to us that frequency domain FLIM would be possible. So we will discuss the frequency domain FLIM in the next lecture, but for the moment we're going to discuss time domain. So how do you do a lifetime imaging in the time domain? Basically, you have to do the standard experiment of time-correlated photon counting and measure the intensity decay in each pixel of your array detector, be it CCD or CMOS. So you have to measure all of these decays and analyze them, and then you use the lifetime numbers to create an image with contrast, which is based on the lifetime being long or short. Sometimes these images can be, I think, get confused because many people add intensity weighting to improve the appearance of the color image, and that's something that confuses me sometimes. The reason lifetime imaging by time-correlated photon counting took off so fast was that all of the components were available in 1990. They just hadn't been put together yet into FLIM instruments. As I recall, the first FLIM instruments were being sold in around 1993 or 94, depending which company you're looking at. But many labs had laser scanning confocal microscopes, and those were the perfect microscopes for doing FLIM. FLIM is really a very generic method. It can be performed on slides, it can be performed on microchannel plates, and it doesn't matter how the scanning is done. The scanning can be done by moving the stage around or by using mirrors to scan the laser beam. So all of these components were already available. And another important thing that was available was confocal detection, because confocal detection helps a lot in improving the resolution of the image. While you can do FLIM without confocal detection, that's less common. And what confocal detection does is it is a method to remove the out of focus emission. What do I mean by out of focus emission? Microscope objectives have a very small depth of field. They're only in focus at a very thin layer, and everything above and below that layer are out of focus, and that gives you a blur in the image because you're not seeing what's at the focal plane, you're seeing through the whole cell. But the optical apertures and the confocal instruments get rid of that. So here's a non-confocal image, and you can see a great increase in resolution, information content with the confocal image because the outer plane fluorescence has been removed. Oh, and I just wanted to show you a couple of other examples because you get entranced by them when you start going on the web and you see how much more detail you can get from a confocal image than you can from a wide field image. Now here's the other things that were already available for lifetime imaging. Previously, we were working with large frame lasers. I mentioned earlier on in the course the difficulties with that and managing the pulse rates and everything. But pulsed laser diodes and pulsed LEDs had become available. There had been great improvements in single photon detectors. The electronics for time-correlated single photon counting were already highly developed, so all that was available. Software. If you've ever worked on a new project where there's no software, it's just a mess because you have to write all the software yourself. But a lot of this software was available. And for any microscope you have, there were several companies who will sell you a kit. Say, all right, I have an Olympus microscope. We'll send you a FLIM kit that you can add on to the microscope and make it into a FLIM microscope. And there were multiple vendors that provided all of these components. So let's look at some examples of FLIM images. You might remember from an early lecture that the Bo-Dippy probes were viscosity probes. We called them molecular rotors because when in low viscosity, they would rotate and the lifetime would drop. In higher viscosities, they would not rotate as much and the lifetime would be longer. So here's a microviscosity image of a HeLa cell done by FLIM. So it's color coded rather than intensity coded. And this part, these bright spots, are a shorter lifetime than the other spots on the cell. So basically, we have, I guess I didn't magnify that image quite enough. But basically, we could say that the short lifetime components are the lower viscosity components and a shorter Bo-Dippy lifetime. That's sort of the small peak down here. And there's a lot more pixels without any Bo-Dippy in them, and they had a longer lifetime, and hence you got a microviscosity image of the cell. This is a pretty neat application of FLIM since, well, some of you listening probably haven't thought about this yet, but as you're getting older, we're all getting these amyloid plaques in our brain tissue, and the surgeons who remove tumors from the brain have learned that by pre-treating the individual with aminolevic acid, they could get some enhanced fluorescence from the tumorous parts of the brain tissue than from the non-tumorous parts. So what happens, this protoporphyrin accumulates because the drug that they used was a precursor for that. The cell didn't continue to put an iron in the porphyrin, so the higher concentration of this compound is shown by the red in the shorter lifetime. And these are also smaller plaques and they're longer lifetimes. But this, if you just did a white light image of this, you probably would see virtually nothing, which is why the surgeons started to use this protoporphyrin method. There are options available with time-correlated photon counting that are not available with almost any other method, but probably not available with frequency domain measurements also. I remember years ago when we were starting FRED experiments trying to do distance distributions, and the analysis was just never working out. We couldn't figure out what was going on. And we finally figured out that we did not have pure DA pairs, donor-acceptor pairs in the samples, but rather we had some amount of donors that lacked an acceptor. So this really confused us for a while, and we had to purify up the samples. With the time-correlated single photon counting and the available of many different laser wavelengths from the pulse laser diodes, you can do experiments in which you measure only the DA pairs, and you can reject the information from those molecules that don't have an acceptor. So the donor or the acceptor might be close or might be further away, but it's still going to be there. So it should be visible if you give a second longer wavelength pulse just to check if the acceptor is there. So this is exactly what's being done by one of the companies. So they get their donor, they know they have a donor photon, then they look and see if there's an acceptor there, so they put in a longer wavelength pulse, and if there's no acceptor, they don't count that as one of the fret measurement pairs, so you could reject those acceptors, those donors that didn't have an acceptor nearby. Remarkably, you can even do fluorescence lifetime imaging with single molecules. Now, in this case, it's a wide field measurement. The reason this was possible to be a wide field measurement is that all the fluorophores were on a slide, and because they're on a slide, they're in exactly the same focal plane, so you didn't have to worry about out of focus fluorescence. You couldn't have done this experiment in a cell with wide field optics because there would be too many fluorophores that were out of the focal plane. So what do we have here? First, we've done FLIM images of single molecules all of the same type, and then we did it with 3 different types of single molecules. And what we have first are the intensity images, and over here at this side are the intensity decays of the single molecules. And of course, they're not going to be real good. Not much signal, they're highly variable. This center part is pretty interesting because this tells you how photostable the molecules are. So these are the time traces from that particular molecule as it's being illuminated, and you can see the CY5 was quite photostable, where the anode dye was blinking on and off and not very photostable. Okay, so the next thing they did is they collected all of these decays together, and they got the decays of each of the single molecules alone. So they have lifetimes. I think I have them. Oh, they're in small print. I have the larger print on the next slide. So we know what the lifetimes are, and then you can make up a histogram for each of the decay times. Now this was not really resolving the molecules from each other because they were on separate slides, but on the next slide, all 3 of the fluorophores were put on the same slide, and then they prepared a histogram of all of the different lifetimes for each of the dots that they measured, and lo and behold, they got a histogram with 3 different lifetimes, and those lifetimes agreed with those expected for the 3 molecules that were used, the CY5 and the atom molecules. So I always thought that was a nice experiment. One of the problems with time-correlated single photon counting is photon overlap or pulse overlap. We discussed this also in an earlier lecture. In order to get a representation of a time-resolved decay, you can only collect 1 photon no more than one photon per laser pulse, because if you collect two photons, well, the instrument detects the start of the experiment, then it detects the first photon, and the instrument is essentially dead when the second photon arrives. It's called pulse pileup. So you've lost that information, so you only see the first photon. So you have to avoid pulse pileup in these measurements, and as you can see, since we want to do so many pixels, you want to collect as many photons as possible, but you're going to be troubled by pulse pileup. This slide I showed you in the previous lectures, where we looked at the intensity decay, number of photons per pulse, and even if you're at 30% or 0.3 photons per pulse, you've already seriously distorted your lifetime. So there are, in some of the company literature, there are claims that you can collect more than 1 photon per 100 laser pulses, but I guess you would have to prove it to yourself that that was working. Or at least I would have to prove it to myself that that was working before I would accept it, because I've seen so much pulse overlap in other experiments. This problem creates pulse pileup creates an enormous problem for FLIM, because now you're trying to collect a 250,000 intensity decays, and I made some assumptions here, so I just did a guess how long it will take to do a scan. So let's assume we're using a 10 megahertz pulse rate. If we can count at 1%, that's 100 kilohertz photon rate is the maximum we can collect. We have this many pixels to fill up, and let's assume we want 10,000 photons per pixel. Most array detectors will hold that many. So we go through those calculations, and we find that it takes, at least half a minute under the best of circumstances to get a lifetime image. And this is simply too long for many biological experiments. So the researchers reduce the number of photons per pixel considerably, so they can get images in less than a second or so. And of course, then you can't really do the multi-exponential analysis that you're used to doing with a cuvette sample. The companies are working on and continuing to work on ways to get more photons per pulse. One way of doing this is to use multiple detectors and routing electronics. So basically, a photon gets detected, and that signal gets sent to one of the counting circuits, and if another photon gets in, it gets sent to another counting circuits. So you can collect the data faster with multiple detectors. And also, there's work being done to reduce the dead time. And you can reduce the dead time between collecting the photon and while the detector's inactive, because it's recording the time, and in the computer you can collect the images faster. So this will continue to improve as time goes on. Here's one I mentioned earlier, the energy transfer experiment with two different color lasers. Here's an experiment that was done with two laser sources, and this was done with biology in mind. This is going to be a cell experiment, and it was excited at two different wavelengths. 370 was chosen to excite NADH and NADPH. 410 was chosen to excite flavin adenine dinucleotide. Of course, now you need a couple of light sources, and you need a couple of circuits to measure the different color photons. And I think this experiment was quite complicated, but it was based on the different photophysics that NADH has and FAD have when they're bound to a protein. NADH can be excited at 350 and it emits out at 450 or so. So we have that laser, and the FAD can be excited at 410. And both of these compounds have fast and slow decay components, whether they're bound or unbound. And in order to keep this discussion simple, I'm not going to distinguish too much between them, except to say, these molecules are in the cell, and each of them has a short lifetime and a long lifetime. And what these researchers did is they looked at normal cells and tumor cells, and they extracted from that data. So they did a multi-exponential analysis at each pixel, and they extracted the fraction of the slow component in the case of NADPH. So these are the images of the intensities or the amounts of the slow component, which is higher in the tumor cell. And in the case of the NAFAD, they put the fraction of the fast component, and that was higher in the normal cell than in the tumor cell. So you can see there's a lot of information that you can get from a lifetime image that you could never see from a white light image or even from just a steady state image of the sample. And of course, this can be expanded. I remember many years ago a presentation at the Biophysical Society when one of the first persons did this, where they hooked a whole bunch of electronics together so they could do multiple wavelengths. But this is now a common feature you can get from the companies, so that the photons are spread out. The light is sent into different detectors. They're sorted. So you are getting not only the lifetimes in each pixel, but you're getting the emission spectra at the same time. Now here's a pretty damning statement. FLIM by time-correlated single photon counting is time-consuming and wasteful. Why do I? And I'm not trying to beat up one of the companies, but this is just the way it is. You can only detect a small percent of the photons. You have a lot of pixels to measure, and the detectors that are using can hold probably, it depends on the size, the pixel size, but 10,000 photoelectrons can be held in a single well. But you're never going to do this in your collecting your FLIM image, because you're never going to detect up to 10,000 photons per image. So all of this storage space is really being wasted, because you're only putting in a couple, let's say tens or hundreds of photoelectrons in a pixel. So because of this, even though time-correlated single photon counting started first, in the literature we can see that other methods are starting to be considered. And these were used initially, but first I'll tell you what it is. It's a time-gated method. So instead of trying to count a single photon, you put in a pulse, and then you take a reading over a short period of time with your detector. This one's easy to understand at the beginning. So you have this gate. It sees some fraction of a nanosecond, and it collects all the light there, and you keep shifting the gate along the decay curve from repeated experiments, and you can collect thousands of photons per pixel this way, because you're not limited to this single photon counting. And just like with the phototube, in the steady state, you can see many more photons than you can see when you're photon counting. And some people would even take it one step further and just measure 2 time bins to get a rough estimate of the lifetime. So the cameras are getting better now. Usually there was no multi-exponential analysis. That cameras are probably getting good enough. We'll probably start to see even more complex analysis with camera gating. So what is the summary so far on lifetime imaging? FLIM is becoming increasing and valuable. It's generic. It can be used with any type of microscope. It can be used with cells or micro channel plates. Software is available. Detectors are advancing rapidly, and it can be formed with other methods which we haven't discussed, such as total refraction, which gives you the equivalent of only seeing the molecules that are very close to the slide, or light sheet super resolution methods. FLIM can be done with any of these. And a surprise to me is there seems to be some movement back to the frequency domain method to increase the rate of photon detection. The reason FD has not been used very much so far is because the CMOS detectors, they're really static detectors, but methods are being developed so you can collect images over short periods of time, and we'll discuss those in the next section. So that's the end of our first discussion of fluorescence lifetime imaging. Thank you for your attention, and we'll see you next time.